All right, so we've talked about the way that we activate sugars, and we talked about how we make glycoproteins. So here we're going to discuss how glycolipids are made. And so here we're going to talk about the structure and function of glycolipids, how they're formed, and what happens when you have defects in breaking down the glycolipids, as well as the importance of glycolipids in the red blood cell antigens. We're also going to talk about what a lectin is, which is a ligand. So glycolipids are basically lipids with carbohydrates attached, just like glycoproteins were proteins with carbohydrates attached. Now, glycolipids are going to be important for us on the cell surface. Again, similar to glycoproteins, they are going to be unique for molecules, so you can recognize different cell types, different tissues, communication, that all that goes into play. The glycolipids are going to be a particular class of sphingolipids, and so these are going to then be synthesized from nucleotide sugars. And again, you're going to add these sugars to a hydroxy methyl group of the lipid ceramide. Glycolipids are going to contain branches often of sialic acid, and then these again are going to be on the membrane cell recognition factors. Sialic acid brings negative charges so that the lipids can interact with the aqueous environment. All right, so let's talk about glycolipids. We're going to have a few terms that are going to pop up. So we have lipid sphingosine, and glycolipids are all going to be derivatives of this backbone. So remember, in our membrane lipids, the backbone is a glycerol. So here it's different as a sphingosine. These are going to develop into cerebrosides and gangliosides. Gangliosides are going to be important. They're going to have NANA, for example, attached to them, sialic acid. So our ganglioside will have sialic acid. They're all going to contain ceramide, and ceramide is basically a fatty acid plus a sphingosine. Sphingosine is going to be unsaturated fatty acid, and these again are going to be important for signaling for differentiation, proliferation, etc. Our cerebrosides are synthesized from ceramide and UDP glucose or UDP galactose, and they are going to contain a single sugar. So here's a galactose cerebroside, has ceramide plus galactose, for example. Gangliosides are going to be a little bit more elaborate, UDP sugars and CMP NANA. So a cerebroside is going to be found in the brain tissue as well as the sheaths of nerves, in the medullary sheaths of nerves. And you're going to break them down, and this will yield a sphingosine, galactose, or other sugars, and a fatty acid. And a sphingosine is a basic unsaturated amino alcohol, which I'll talk about in just a moment. We have neuramic acid, which is a version of sialic acid, and acetylneuramic acid is important for cell adhesion and migration, and these are going to be our gangliosides. All right, so what does all this mean? So let's just break this down just a little bit into something we can sort of digest a bit more. So we know that we have membrane lipids, and we know we have a fat, and a fat has a glycerol backbone, right? And it doesn't matter what kind of fat it is. If it's a phosphatidylcholine, phosphatidylethylene, you're always going to have that glycerol backbone. The difference between a stored fat, which would be our tag, and our membrane fats, which would be like a phosphatidylcholine, is the addition of a phosphate on that carbon 3, which is basically phosphatidic acid. Now, when we're talking about glycosylation, though, we're talking about our ceramide backbones. And so let's discuss this a little bit. So here we have a sphingosine backbone. And basically we have this carbon again, and we have one fat attached to that carbon, which is a carbon 16, so that R group is 16 carbons long. And we have a nitrogen. And so that's very different than our glycerol backbone, right? So here's our glycerol backbone, and here's our sphingosine backbone. Now, when we have ceramide, we're basically taking the sphingosine backbone, which is this purple here, and we're just adding another R group. And ceramide's going to add another fat to it. So basically, ceramide is sphingosine, which has one fat group already. I'll make the fat groups in green. and it's adding a second fat group to that nitrogen. So now we have two fats, and that's ceramide. Now, sphingomyelin is basically going to take that same ceramide backbone, which is the purple and the blue, and it's going to add a phosphate group, and then you're going to add a phosphoethylamine group, and you're going to generate sphingomyelin. And a cerebroside is you're going to take that ceramide backbone, and then you're going to add another sugar to that group, and you're going to generate a cerebroside. And then when you take and make a ganglioside is, again, you have that ceramide backbone here, you have your cerebroside attached to it, and now you're going to get more attachments to it, including eventually a sialic acid residue attached to it as well. So basically, with these glycosylated fats, we're adding sugars, remember? You're starting with your ceramide backbone, because your ceramide backbone is going to get glycosylated. In a cerebroside, you glycosylate your ceramide backbone. In your ganglioside, you're adding even more sugars to your ceramide backbone. Okay, so that's what those terms mean. Now, the importance of glycolipids are for communication. We have our red blood cell antigens, ABO antigens. Glycolipids also are important for absorption of nutrients, receptors for microbes. Again, they can bind to our glycolipids, our sugar residues. ABO antigens, what is an antigen? An antigen is basically any molecule that you can make an antibody to. So our ABO blood group antigens are going to be actually the sugars added to the glycolipids on the surface of cell. And also carbohydrate portions of serum glycoproteins as well. So the ABO is on the surface of the red blood cell, and they are going to be linked to those sphingolipids, and so they're glycosphingolipids. Basically, remember we just talked about what a sphingolipid was, and you're just adding sugars to it. Your ABO in the serum, meaning that it's not attached to the cell but is free floating in your blood, are carbohydrates associated with proteins in the form of glycoproteins. Okay, so just remember that ABO is referring to the sugars. The sugars are going to be in a certain conformation attached to the surface of a red blood cell bound to lipids, or the same ABO sugars are going to be floating in the blood attached to proteins. It's important to note that not everybody secretes the ABO antigens or sugars. So you have secretors versus non-secretors, and this can also be used in forensics. All right, so basically we have our sugars, and so you can see that these are the sugar, and this R is either going to be a lipid or a protein. Okay, so that R can be either lipid or protein. So the structures of our blood types, are type O, type A, and type B, are the same except for this end sugar here. So you can see they all have N-acetylglucosamine, they all have galactose, they all have a fructose. So that is the same. The difference is in type O, you have no sugar attached at this outer portion. In type A, you're going to have another well, in this case, it's a galactosamine, N-acetylgalactosamine. And then in type B, you're going to have just a galactose. And this is all it takes to get rejection if you mix up the blood types. Now, we can have defects similar to what we saw with our proteins if we have lysosomal deficiencies. Your lipids are also going to traffic through lysosomes, and lysosomes are going to have hydrolases that are going to remove the sugars. And these are going to remove the sugars from both proteins and from our lipids. And so we have our glycosidases, exoglycosidases from non-reducing ends are very specific. We have endoglycosidases is going to cleave carbohydrates within and have broader specificities. So there are lysosomal storage diseases that are inherited, and this is basically you have a defect in enzymes that remove the sugars. So here's a list of defective enzymes. These are all referred to as gangliosidosis. You're going to have a defect in removing fructose. You can have a defect of hexamidase, which the lipid you can see accumulates here. You have a ganglioside that's going to be accumulating a GM2 ganglioside versus a GM1 ganglioside, and they're just going to be different in their structure, their sugar structures. But basically, in all of these diseases, you're going to have an increase in your products. So here we're showing you have choline, sphingomyelin, and this is in Niemann-Pick disease. Niemann-Pick disease is a disease where children actually self-mutilate themselves. We have Farber disease, Gaucher disease, Tay-Sachs disease, etc. And all of these have to do with an accumulation of lipids because you don't have the enzymes to remove the sugars from those lipids. Now, lastly, we want to just talk about lectins. Lectins are going to be proteins and recognize carbohydrate structures. So the lectin itself is a protein, and it's going to recognize carbohydrates. They are all going to contain what is called a carbohydrate recognition domain, or a CARD domain. And so a lot of times your pathogens, viruses, bacteria will recognize carbohydrates. They'll have lectins associated with them. For us, we do have our own series of lectins. We have a lectin that's called C-reactive protein. This is going to be important in activating complement, for example. We also have different series of selectins, and we have a mannose binding protein. All of these are involved in the immune system and in protection against potential pathogens, for example. This is just an example of a lectin binding to the RBO antigen and agglutinating the red blood cells. Okay, so just keep in mind glycolipids. We have sphingolipids that add carbohydrates to that ceramide backbone, not the glycerol backbone. Sialic acid is going to be formed in the cytoplasm, activating the nucleus. We talked about that in the previous lecture. We're going to have different enzymes that are going to have defects, and that can lead to an increase in glycoproteins or glycolipids, and so you have different diseases associated with deficiencies of those enzymes. And then finally, lectins are proteins that are going to have specificity in binding to the carbohydrates that are coating either our proteins or our fats, our lipids.